Okay, let's start. Chromosomal theory of inheritance. Here, Mendel published his work on inheritance of characters in 1865. For several reasons, Darwin is also one of the reasons for that. It remained unrecognized till 1900. These are also going to be questioned here, 1865 and 1900. Okay, 1865 he published his work, but nobody noticed, nobody has taken care about this particular. Moving on, so communication was not easy in those days and his work could not be widely publicized. One of the reason they are saying. Secondly, his concept of genes or factor, actually he said it as factor. In Mendel word, factor is a word. As stable and discrete units that control the expression of traits and of the pair of allele which did not blend with each other was not accepted by his contemporaries as an explanation for the apparently continuous variation seen in nature. And whatever he said in the theory of principle of inheritance, what he said, stable and discrete unit. He said factors as stable and discrete unit. So-called factor nowadays, we call it as gene, is according to him, that is factor, stable and discrete. This is the word, in the words, in the belief of Mendel, in the factor is stable and discrete unit that control the expression of trait. Control expression of different characters, okay, and one another is pair of allele did not blend. According to Mendel, pair of alleles did not blend. What is blending? When you cross a red flower with white flower, if pink flower comes, that you call blending. So these three, that's the factors, gene, stable and discrete, controlled expression, pair of allele did not blend. These facts, whatever the people who are there, contemporaries at the time, is not accepted. So, the second reason. First reason, not widely publicized. Darwin is famous at the time. And people are busy with whether even we came from monkey or not. At the time, people are not much focused on these theories of inheritance. And what is the third one? Contemporaries as an explanation for the apparently continuous variation seen in nature. Then, thirdly, and according to them, there is continuous variation. So, how can you say stable and discrete factors are present? That is their approach. Thirdly, Mendel's approach of using mathematics. He used mathematics. He also graduated in physics. So that's why he's good enough in maths and he explained all these biological phenomena in used mathematics in this biological phenomena and most people didn't understand this kind of Mendel's mathematical approach and the theories of inheritance. That is also one of the reason we can say. Okay, so phenomena totally new and unacceptable to many of the biologists of his time. Finally, though Mendel work is suggested that factors were discrete units, he could not provide any physical proof for the existence of the factors or say that they were made up of. So, no physical proof. What is physical proof? After that, you learn in molecular basis of inheritance, the physical proof. So many scientists made experiments like Griffith transformation principle, Avery, MacLeod, McCarthy experiment, Hershey and Chase experiment finally proved that the physical proof. What is the physical proof? DNA, nucleic acid, RNA. So these are responsible for the formation of the so-called genes and factor, which Mendel definitely don't have idea on these. So they don't know the DNA and RNA. So that's why we can say Mendel is not able to show the physical proof. At least finally we proved that Watson and Crick, so many scientists' efforts finally came for the physical proof, but in 1865, when he submitted, these are the problem. One is not widely publicized. And so-called factors, nowadays we call gene. Gene is not the word used by Mendel. Genes proposed by Johansen. But for your understanding, every time wherever factor is there, the gene name is also given. So factor, stable and discrete units, he said, stable and discrete unit, controlled expression, pair of allele did not blend. All these not acceptable by most of the scientists of the time. And he used mathematics in biological phenomena. Most of the people don't know that. That application of mathematics, probability mass. He used probability actually mostly in his theories. Probability laws are there, but people are unaware of biology. People are unaware of that particular kind of phenomena. So no physical proof, so-called DNA and RNA at the time, not in the use or not yet discovered at the time, we can say. No physical proof is shown over. He said factor. So where is the factor? Now we have answer. Where is the factor? It is there in the nucleus, in the chromosome, which is made up of DNA. We have answer, but at the time we don't have answer for that. Okay. So moving on. So till here, that Mendel experiments we are talking about. In 1900, the three scientists, who are they? De Vries, Karel Karens, Eric van Scheermark, Hugo De Vries, the Karel Karens, and Eric van Scheermark. These three are said to be rediscoverers of Mendelism in 1900. They rediscovered Mendel's result on the inheritance of character. Also, by this time, due to advancements in microscopy that were taking place, scientists were able to carefully observe cell division. So, a complete idea on the cell division also came, and this is one of the criteria. This led to the discovery of structures in the nucleus that appeared to double and divide just before each cell division, and they named it as chromosomes. Hofmeister is the scientist who proposed the chromosomes. Colored bodies, they are visualized by staining. When you stain, they appear with one color. So by 1902, the chromosome movement during meiosis had been worked out. The movement of chromosomes during poles at anaphase, these kind of things are worked out. Then Walter Sutton and Boveri, those scientists who are here I am writing, and those scientists who help behind the screen, that means behind our textbook, I am just saying, I am not writing. Because we don't get that kind of questions. Like Hofmeister, chromosome, Johansen, gene. So Walter Sutton and Theodore Boveri noted that behavior of chromosome was parallel to the behavior of genes. What is this word? Chromosomal behavior. Chromosomal behavior is parallel to the behavior of genes. Parallel to behavior of genes. Okay, so this is the word chromosomal. Why? If it is assertion, chromosomal behavior is parallel to the behavior of gene. If it is assertion, what is the reason? Because what will be the reason here? Genes are located on chromosomes in certain loci. Genes are located on chromosomes in certain loci. Okay, so this is the paragraph. In this paragraph, 1900, Hugo De Vries, Karel Karens, Eric van Scheermark is the scientist who rediscovered the Mendelism. And after that, we have some investigations like chromosome and genes and the various other investigations like the cell division process and how the chromosomes move, and along with the chromosomes, whatever the genes present on the chromosomes which move towards the pole, all these particular patterns came under limelight, and then it makes Mendel more understandable. Okay, the two alleles of a gene pair. Now let's talk about this. The two alleles of a gene pair located on homologous sides of homologous chromosome. What is this homologous chromosome? What is homologous chromosome? In every pair, human being is having 23 pairs of chromosome. in every pair of chromosome, in every pair, one chromosome you get from your mother and one chromosome you get from your father. 23 pairs of chromosome. Every chromosome. One is from mother and one is from father. Like if it is from mother, we call this kind of chromosome as maternal chromosome. And a chromosome you got from your father called paternal chromosome. Okay, these two chromosome pair is called as homologous pair of chromosome. Homologous pair of chromosome. If we talk about blood groups, certain locations, in one certain location, if I say A blood group, gene for A blood group, IA, if it is present, in the same location, the respective allele, whatever, IO or small I, whatever, that respective allele. So, what are the alleles for the blood group? A, B, and O. In the form of genes, if you want to show IA, IB, and I. So, try to understand the difference between homologous pair of chromosome and allele. Okay, now let me talk about one more word here, non-allele. For example, in the same chromosome, in another place, what we'll take, let's take height, capital T. Their allele, the respective allele will be small t. Okay, so I took the character of height, not a single gene represent height in case of human being. Just for example, I am taking. Okay, so what is A for this T? Non-allele. So try to understand this pattern. Try to understand. We'll go to the board and let us see once again what is allele, what is non-allele, what is homologous pair, what is non-homologous pair. If you understand this, you can understand the text lines easily and comfortably. Okay, so first of all, let's talk about the homologous pair. So such kind of pairs, 23 pairs are present in human being. One you get from mother, one you get from father. Maternal and paternal. In certain loci, for a character, for blood group I am taking, capital A and small I. That means this blood group is going to be AO. In the same chromosome, in other place, in another location, I'm talking about height. I took a word, a letter, capital T, small t, two alleles on the same location. So you have to find it out. A particular location having a character, similar character, maybe same allele, different alleles can be expected over. Okay, can be expected over. So let me write one more here, one more like capital S and small s. Some other character we are writing here, some other character. Okay, so what is A to the S? This is non-allele. Non-allele. What is A to the S? That is non-allele. That is very important to understand. And what is A to the O blood group? Allele. Okay. So what is this? Homologous pair of chromosome. This diagram, this alone single diagram will be much helpful for us to understand the homologous pair of chromosomes, non-homologous pair. Okay, this is the 9th chromosome. Let me draw the 10th chromosome for you. 10th chromosome. So I am drawing 10th chromosome. Such kind 23 pairs of chromosomes are present in the human being. This is again maternal and this is paternal from mother and from father. Okay. So they may also have certain genes. Every chromosome will have some genes. We have 23 pairs of chromosome, 23 haploid set, 30,000 genes. Average on the chromosome you might find 3,000 genes. So that's why drawing 2 or 3 or 4 is not at all a bigger matter. So average, that means for example on first chromosome we have 2,968 genes are there. We learned in Human Genome Project. 2,968 genes are present on first chromosome. Okay, So what do you say that particular genes? See this? This is allele. Some capital F, small f. Some capital G, whatever it is. Either dominant form or recessive form. Capital F to the G is non-allele. Capital F to the small f is going to be allele. Okay, allele. So now with these, we'll frame some some questions, some statements here. This red one to this black one or red one, whatever. What do you say it as? Non-homologous chromosome. Non-homologous chromosome. That means a chromosome of 9th to any of the chromosome of 10th or 11th or 20th or 23rd, whatever it is, we can say it as non-homologous pair of chromosome. Okay, non-homologous pair. Okay, during meiosis, from here, from these two chromosomes, we get 1. From these two chromosomes, we get 1. Okay, so these two can be called as non-homologous chromosome. If I say like this, we have 23 pairs of chromosome. Among the 23 pairs of chromosome, some 23 chromosomes are present in the sperm. These 23 who are present in the sperm are non-homologous chromosome. You need to understand these phenomena. Then 23 pairs in the ovum are going to be non-homologous pair of chromosome. First, if you understand this language, then we can go to the textbook to understand that statements or lines, whatever there in the textbook. Okay, moving on. Homologous chromosomes. Alleles, we use that word. Allele, non-allele. Okay, so here only, before I go to the text, here only I'll frame that line. Non-allelic genes which are present on one chromosome. See that line which I am drawing? I'll draw a little thicker to understand this much better. Pink line, this chromosome. So on one particular chromosome, the total number of genes, non-allelic genes, we said these genes are called non-allelic genes because they represent different character. Non-allelic genes present on one chromosome or same chromosome, one or same, whatever, are called linkage. Non-allelic gene present on one chromosome is called linkage. If you understand, linkage is a bigger topic. It was a little, 1% is given in our text. Think little wider, you understand much better this. Okay, means they explain about two genes which are present on a chromosome. But I said to you, there are 2968 genes are present on one chromosome. Then means here if I draw this, I have to draw 2968 locations. All the genes present on one chromosome are called as linked genes. And the phenomena is called linkage. Phenomena is called linkage. When you move this chromosome to the gamete, along with that chromosome, all the genes also will come. This is the criteria. During cell division, during anaphase, that chromosomes are pulled towards the pole by spindle fiber, isn't it? So when you move a chromosome, all that linked genes which are present on the same chromosome are going to be enter into that particular place where the chromosome is getting pulled over. Okay, moving on. Let's try to understand that text lines now. The important things to remember that chromosomes, that chromosome as well as genes occur in pair as I shown to you. Two alleles of a gene pair are located on homologous sites on homologous chromosome as I shown to you now. So let me show you. Two alleles of a gene are present on located on homologous sites of location on homologous pair of chromosome. Just I am visualizing whatever the line is given here. I am visualizing homologous pair of chromosome, homologous alleles. Sorry, what I mean, as well as genes in text language, two alleles. So this is the paragraph, and moving on. So comparison between the behavior of chromosomes and genes. Okay, let me show you here again. This is the chromosome and above on the chromosome. It's just like a comparison between the bench and students on the bench that you need to understand in that particular pattern. Chromosome is the bench, and certain locations having certain genes are all going to be the students who are sitting on the bench. Okay, moving on. Chromosome occur in pairs. Okay, I've just shown the pair, one pair, homologous pair to you. Okay, genes are also occur in the pair. I am just pointing out this is genes which are in pair. Segregate at the time of gamete formation. As I said to you, they will be pulled towards the poles during anaphase of the cell division, such that only one of each pair is transmitted to a gamete. Means either which one chromosome reaches to one pole and other to the pole. If we talk about human being, we have such kind 23 pairs. So 23 in the sperm, and you'll find 23 in another sperm. So in that way, ovum 23 out of 23 pairs, 23 in the ovum. That is called single segregation. Independent pairs of segregate independently of each other. Okay, independent pairs segregate independently of each other. So I just shown one particular color only to you. Now I am taking another pair of chromosome. I can't show the same phenomena with 23 pairs of chromosome, but at least I can try for you with two pairs. I just am taking two colors here again. Two color. So black and blue. These two are another homologous pair. The first pair, and this is the second pair. Imagine there are two pairs of chromosomes in the particular organism. So how will it happen? Okay. So similarly, these are also going to be pulled to the poles, isn't it? So how it will be? How it will be in the gamete? In the gamete. So four possibilities are there. If I am taking two, four possibility. From these two pairs, one I took, and the another one I should take. Another one I should take black. This is the gamete. From these two, I took red. From these two, I took black. We call them as non-homologous. Out of two pairs, two came. Out of 23 pairs, 23 will come. So two pairs, two came. One is red and other is from these two, we got black. Next, what are the other possibilities? For the same red, you might get blue also. You might get blue also. These are not homologous pairs. Formation of gametes I am talking about. I am trying to explain in my way. That diagram is given in textbook even. Now I am taking green. For this green, there is a possibility of, for this green, there is a possibility of black, and there is a possibility of blue. These are the four possible combinations are possible. Okay. So red and black, red and black, red and blue, green and black, green and blue. What do you say this kind of phenomena? This kind of phenomena is called as independent assortation. Remember this. This is called independent assortation. During the formation of gametes, Mendel more focused on this actually, independent assortation of gametes. Whatever he said over, independent assortation of gametes. Okay. So, possible combinations, possible formation of gametes. Four possible combination of gametes will come here. Now we try to read this. Independent pairs segregate independently each other. Okay. Moving on. One pair segregate independently of another pair. One pair segregate independently of another pair. This is true for the genes which are present on different chromosomes. One pair do not segregate if they are present on the same chromosome, but one pair, if genes on one chromosome, one pair of genes on other chromosome, if you take, it will be true. That is the confusion actually students will get in this area. Okay, moving on. During anaphase of meiosis 1, the two chromosome pairs can align at the metaphase plate. That at least I am just showing that metaphase plate only to you. Okay, to understand this, compare the chromosome of 4 different color in the left and right columns. So I just shown the colors here on your left side. In the left column, okay, whatever they said, their colors they used over and I use my colors. Homologous pairs, one pair I just shown red and green, other with black and blue. And how they get independently assortated, I just take over. Now, let me write the same phenomena here. Here also, you try to understand these particular lines. They took homologous pair of chromosomes at metaphase. Why it is little confusing, what is the difference between the diagram I draw and there? They are actually true in that way. Why? Because during S phase, chromosomes are going to be replicated, isn't it? Replicated. That's why I have to show like this. I just shown for easy understanding like this. Single chromatid I just shown. Okay, they shown in this particular pattern. That is the only difference actually. That is the only difference. So if the particular chromosome is double, let me show you here. I took red and green, isn't it? Red and green. So for easy understanding, I just took like this. What's the problem here? That may not match with the original because that is chromosome are going to be replicated during S phase. Cell division law, S phase. So that is the thing, only the difference. But it is clearly understandable. Once you understand like this, even you understand like this. So that is the reason I just shown in that way. Okay. Sutton and Boveri argued that pairing and segregation of a pair of chromosomes would lead to the segregation of a pair of factors they carry. What is the principle here? Chromosomal theory that argued that pairing or separation of a pair of chromosomes, when a particular pair of chromosomes are segregated towards the poles during anaphase, that also the genes present on that chromosome are also segregated. So I am saying if I separate two benches, the students which are present on the two different benches are also getting separated. In that way I am talking about. Okay, you just compare the chromosome as a bench and the students on the bench are genes. That you understand much better. Then Sutton united the knowledge of chromosomal segregation with Mendelian principles and called chromosomal theory of inheritance. Actually, Mendel theories are more understandable after chromosomal theory of inheritance, which is proposed by Sutton and Boveri only. Following this synthesis of ideas, experimental verification of the chromosomal theory of inheritance was done by Thomas Hunt Morgan. Is another scientist who is coming here. Sutton and Boveri came. Thomas Hunt Morgan. And Morgan worked with tiny fruit flies, Drosophila melanogaster, which is considered as Cinderella of genetics, very important. Most of the experiments has done on Drosophila only, which were found very suitable for such studies. They could be grown on simple synthetic medium in the laboratory. They complete their life cycle about 2 weeks. That is a very interesting thing which makes us to understand the generations in a short period of time. And a single mating could produce a large number which is required for the experiments here. We should see the flock in huge number that you can understand the patterns of inheritance easily. Okay, so these are questions here. Lifespan is about 2 weeks. Okay, Drosophila melanogaster. Morgan worked on them. A single mating can give more number of progeny. Also, there was a clear differentiation of the sexes, male, female, easily recognizable. You see here, the short one is male and that large size is the female. You can see in the flag who is male, who is female easily. That is also one of the reasons why we select Mendel. Similarly, like why we select Pisum sativum, why Mendel selected Pisum sativum. In the same way, why Morgan selected Drosophila melanogaster for his experiments, Drosophila melanogaster. Reasons they are saying the lifespan is short, they can be grown very easily on the simple synthetic medium, and we can recognize the male and female easily. So, and also has many types of hereditary variation. Drosophila is with full of variation. Red eye, white eye, long wing, vestigial wing, likewise. Okay, so these are the reasons why Morgan selected Drosophila melanogaster. So, see this, we can see this as a statement question. What is the reason Drosophila melanogaster selected by T.H. Morgan for his experiments? Now, I think after all these, we understand the patterns of linkage and patterns of recombination easily. So now let's talk about this. Morgan carried out several dihybrid crosses in Drosophila to study genes that were sex-linked. Okay, so sex-linked. The word is more pronounced for X-linked in the total text. You remember, we have three types, X-linked, Y-linked, XY-linked, but it was more pronounced. Whenever they say sex-linked, you should understand that is X-linked. The crosses were similar to dihybrid as Mendel did, dihybrid cross. That I am showing the Mendel dihybrid cross. For example, Morgan hybridized yellow-bodied, white-eyed female to brown-bodied, red-eyed. Mendel line, dihybrid cross of our so-called Mendel, he's taken two characters, seed color and seed shape, isn't it? Seed color and seed shape. This is seed color, this is seed shape. Seed color, seed shape. So two characters, seed color and seed shape. In the same way, I'm taking Drosophila, yellow-bodied, brown body, means he took two characters, color of body. What are the two contrasting traits for that? Yellow and brown. Okay, yellow and brown. And what is the another one? Eye color. White-eyed and red-eyed. Color of eye is the another character he took. Color of eye, white eye and red eye. Okay, so like seed color and seed shape, yellow, brown colored body, white, red. Color of eye is having two alternative forms. What are they? Yellow and brown. Color of eye is having two alternative forms, two alleles. What do you say? White and red. Now, let us talk about cross, which is given here in the text. Morgan hybridized yellow body, white eye. I am writing here yellow body, white eye. Yellow body, white eye. Okay. Next, yellow body, white eye crossed with brown body, red eye. Brown body, red eye. Similarly, yellow colored seed with round shape, yellow colored seed with round shape, or yellow round with green wrinkle, it's like in that way. Yellow body with red eye. Okay. He observed that the two genes did not segregate independently of each other, and the F2 ratio deviated very significantly from the 9:3:3:1 ratio expected when two genes are independent. Okay. So I will continue. I will explain both independent assortation and this one at a time. So moving on. Morgan and his group knew that genes were located on the X chromosome and saw quickly that when two genes of dihybrid cross situated on the same chromosome, the proportion of parental gene combination were much higher than non-parental type. Okay. So to understand this phenomena, I have to come back again. So let me take this to the page. Let me take this. Okay, let me take another page. Then only we'll understand. I'm going there. I'm bringing that page here. Okay. Let's bring this page. to learn, to explain, to make it much better. So allow them, let it, the page will be here. Now let us try to understand the phenomena. So first of all, let us try to understand what is Mendel's independent assortation. Mendel theory of independent assortation. Then after only we understand these particular lines, whatever shown here. So that's why let me go. So what is the cross he did? He took yellow colored seed with a round shape, green colored seed with a wrinkle shape. Okay, yellow colored seed with round shape, green and wrinkle. Yellow color with round seed, green color with wrinkled seed. We call these combinations are parental combinations. Okay, this is P1 generation, parental generation. Okay, so what we obtain? So for this chromosomal form I am giving here, yellow round crossed with green wrinkle. So what we obtain in F1 generation, heterozygous yellow round. Heterozygous yellow round are appeared in the F1 generation. F1 generation. Then when F1 is self-pollinated, F1 is self-pollinated, in F2 generation, after the Punnett square board, we got 4 combination. Yellow round, green wrinkle, yellow round and green wrinkle, and yellow wrinkle, green round. 4 combinations. The 4 combinations are appeared as in the form of 9:3:3:1 ratio. This is F2 Mendel dihybrid cross. Okay, so in that, let me point out these two. Yellow round. The seed which is yellow in color, round in shape, the seed which is green in color and wrinkle in shape is natural phenomena. What I mean? Parental combinations. What is another one? Yellow round. Okay, so yellow round. These are parental combination. This is the parent and this is the parent. Okay, so what is this then? These two appear who are appeared in the F2 generation. Yellow colored seed in wrinkle shape, green colored seed in round shape. These two are said to be recombinants. Recombinant. Remember this word recombinant. Recombinants are possible. Okay. So independent assortation. That what the word independent assortation, which is shown here, is possible because this chromosome, these genes, where it is present. I am showing this particularly. See my underline. I am showing this. Whatever we learned till the time came to show our knowledge here. How does they appear? I am taking again 4 colors. Okay. 2 pairs of chromosomes. 2 characters, 2 chromosomes we are taking. Okay. Now, let me show that seed color. Where is that seed color? Here on 1 chromosome. Let it be, that is on first chromosome. And where is our seed shape? That is on second chromosome. Okay, now let's go through this. Possible formation of combinations. From these two, one should come. From these two, one should come, isn't it? Then let me draw that. See the colors, whatever I drawn. As I am taking the same mistake again. I didn't duplicate the chromosomes to understand much better. Okay, I am taking green. Non-allelic genes. Okay. Let me draw first of all. Then after I'll use that words, the technical terms we use. Okay, green again. From this side, I took red. Okay. From this one, I have to take. I'm not moving genes. I'm moving chromosomes. I'm moving chromosomes here. Okay. So, next, the green one, orange one I have to take. Red with blue, red with orange. Am I correct? Next, green with blue and green should be with orange. So now let me draw their possible combination. Non-homologous chromosomes these are. From this pair 1 and from this pair 1 we have to take over during the formation of gamete. Okay, red and green are said to be homologous. Blue and orange are said to be homologous pair of chromosomes. Okay. Now, when I pulled the chromosome towards the pole, I have to pull the gene also. This is capital Y and what is on the blue? Capital R. So, for that I will show you the gametes here. Capital Y, small r. Gametes, how it will come? Capital Y, capital R, capital Y, small y, capital R, small y, small r. So, this is the one. Capital Y, capital R. Non-allelic genes present on different non-homologous pair of chromosomes. What is red to green homologous pair? Here, non-homologous pair of chromosomes. What is red to the blue? Non-homologous. This is the word, non-homologous. Seed color on one pair of chromosome, seed shape on another pair of chromosome. Okay. So, non-homologous pair of chromosome. Next, I think you understand the phenomena, what I am doing. Capital Y and small r. This is another gamete. Now small y, green one. Small y and capital R. I am trying to show the same phenomena which we do in this particular table in chromosomal form. Okay, what is the next one? Green one, small y and orange one, small r. So this is the genetic types. Okay. Now, okay, let me go to the next page, and we will bring that text here to understand this phenomena. So paragraph wise we have to bring over. Let me take paragraph wise. One after another, one paragraph each other will bring. Morgan carried out several dihybrid crosses in Drosophila melanogaster were sex-linked. The crosses were similar to dihybrid crosses. So how does the dihybrid crosses are present? I just shown now. Similar to dihybrid crosses that I shown now. How the dihybrid crosses can be crossed, Mendel in peace. For example, Morgan crossed yellow bodied with white eye. Whenever the problem comes, you should write like this first even. To brown bodied with red eye. And intercrossed their F1 progeny. Okay, what is the F1 progeny? He observed that two genes did not segregate independently each other. F2 ratio deviated from significantly 9:3:3:1. And a similar class when they did like Mendel, we got 9:3:3:1. He is saying that kind of ratio not came. What ratio came will come after, but this is the criteria. So if I do like Mendel pea plant, if I do in Drosophila melanogaster, I didn't get same result. We want to say that I didn't get same result. Okay, moving on. We focus on here that you will understand the phenomena only in the end only. So till the time it seems to be little confusing. So next paragraph I am taking. Next paragraph. Till here we'll take. Now, Morgan and his group knew that genes were located on the X chromosome, saw quickly that when the two genes in a dihybrid cross were situated on the same chromosome, the proportion of the parental gene combinations were much higher than the non-parental type. Okay. parental type of combination are more than non-parental type. So, sir, what is parental type? What is non-parental type? Let me show you the same in case of Mendel, and then you understand in this. Yellow round and green wrinkle are parental type. Yellow round and green wrinkle. Yellow wrinkle and green round are non-parental type. You may call it as recombinants. Okay. So, the proportion of parental gene, Morgan attributed this is due to the physical association of the linkage of the two genes, coined the term linkage. Okay. So, for Mendel, whatever we are saying, independent assortation is possible. Why independent assortation is possible? Frequent. Red means red with blue, red with orange, green with blue, green with orange. We call it as independent assortation. So selected two characters, one is seed color and seed shape. Selected two characters located on seed color and seed shape, located on different homologous chromosomes. Different homologous chromosomes. This is one homologous pair and this is another homologous pair. That's why what is possible? Independent assortation is possible. Okay. So now what Morgan want to say is the selected two characters are present on the same chromosome, like seed color and seed shape. If they are present on the same chromosome, when you take the gametes, you will get only two types of gametes. What are they? Parental gametes. Okay, only two types of gametes. Might be there is a crossing over occurs. That's why after crossing over, this piece will cut down here and this piece will be exchanged with them. That's why you get some kind of recombination. But definitely recombinants are less. Definitely recombinants are less. If you notice, 25%, 25%, 25%, 25%. This is parent, this is parent. Parents equal to recombinants. These two are recombinants, gametes. Okay, so when does parent equal to recombinant, parent gametes are equal to recombinant gametes? When the two characters located on two different chromosomes. Percentage of gametes is going to be equal. Parent 50%, recombinants 50%. So Morgan noticed some other phenomena. Parents are more than recombinants. Parents are more than recombinant. More parental gamete, more parental, less recombinants. Why? Why means now we have to say this. Why means linkage. What is linkage? Capital Y, capital R. Both are said to be non-alleles, isn't it? Non-allelic genes located on same chromosome. Non-allelic gene located on same chromosome. Let me continue. I am just following the text here, not in my style I am explaining. I am just following the text here. Morgan attributed this is due to the physical association of the linkage of the two genes and coined the term linkage to describe this physical association of the gene. I'll write that physical association here also. We have a gene, two genes, capital B, small b, capital R, small r. I am showing this is linkage, non-allelic gene present on same chromosome. This is called association of genes on a chromosome, and the term recombination to describe the generation of non-parental. Okay, so we got actually capital B, small b and capital R, small r. Sorry. Okay. What are the other combination we should get actually? Capital B, small r. Wait. We got a small confusion here. So if I explain with the same pattern only, then you understand. Capital Y, small y, capital R, small r. I should not take other letters in this phenomena. So, this is one gamete, capital Y, capital R, parent gamete. This is one gamete, small y, small r, parental gamete. So, when does the recombinants will come? Due to recombination, or you may call it as crossing over. That word crossing over. During crossing over only recombination. This piece exchanged with this piece, capital Y, what will come? Capital Y, small r. Small y, capital R. These two are said to be recombinants. But here in this case, parents are more. These two are the parents are more than recombinants. Why, sir? Why? Because see the difference. See the difference. What's the difference with the previous one? What is the difference with the previous one? Mendel selected two characters present on different chromosome. So if I am asking you, if sir selected two characters present on the same chromosome, what happens? What happens? Parental gametes are more, recombinants are less. We get. Why? Linkage. What is linkage? Two different genes, non-allelic gene present on same chromosome is called as linkage. Okay. Moving on. Morgan and his group also found that even when genes were grouped on the same chromosome, some genes are very tightly linked. So, for that I will show you. Now, I just shown capital Y, capital R. Okay. Imagine if the same is present like this, capital Y, capital R. This line will be much helpful. Some genes are very tightly linked, show very low recombination. Okay, very low recombination means, let me draw their homologous pair also, small y, small r. So if even crossing over occurs, recombination, these two will go at, because distance between the two genes is same, that's why both genes will go, isn't it or not? That's why very less recombinants will come in this condition, because even crossing over may not separate them. While others were loosely linked, this is the word, loosely linked. This is the word, small y, small r. This is loosely linked and this is tightly linked. Sir, can you show me not linked? Yeah, I'll show you. Not linked. Not linked. Not at all linked. Selected two characters present on two different pair of chromosomes. That is not linked. This is loosely linked. This is tightly linked. So what happens not linked if they are present on different chromosome? Parental gametes equals to the recombinant gametes. You get 9:3:3:1 ratio. Sir, what happens if they are loosely linked? Loosely linked. You get some more parental gametes, less recombinants. Loosely linked I am talking. Less recombination is due to crossing over. Then, sir, what happens when they are tightly linked? More and more parents only. Very less recombinants are formed. Why recombinants are very less? First of all, they go their own way. So before they go, if any crossing over occurs, even though crossing over occurs, they may, means there is less possibility crossing over occur here, isn't it? Very closely linked. That's why less recombinants. If they are much closer, no recombinants at all. So this is the criteria. Okay. For that, we found that the genes in yellow were white tightly linked and showed only 1.3% recombination, while white and miniature wing. This phenomena I will show you in the text here. This phenomena, the diagram I will show you here. So if you are able to understand this diagram, that means you understand linkage. First of all, I said about what is linkage. What is linkage? I am taking this. I am bringing this to the, let me cut your permission. Wait. Should cut from here. Okay. Oh, that we have to go another page. Let me cut. Yeah, paper is ready. Now, let us understand this diagram. So here, linkage is explained. One more interesting thing here, it is X linkage. Means there is linkage, okay. But one more confusion here, this is X-linked also. What happens in the X-linked case? You don't find the gene on Y chromosome. You don't find the gene on Y chromosome. That's the problem. Actually, people have more confusion in this area. Till the time I explained what is linkage. Sir, what is linkage? Presence of two different characters on one chromosome is called as linkage. I said it as, isn't it? Present of two genes, two non-allelic genes present on same chromosome is called linkage. Okay. Sir, what is non-linked gene? Non-linked means this is the non-linked. Same two characters present on different. It is Mendel. First of all, you understand this page is Mendel. Okay. Next, after that, we came to the pattern called linkage. Linkage, linkage. This is linkage. This page explains linkage phenomena. Linkage might be genes present on the same chromosome is called linkage. Or genes present on the same chromosome, very close linkage, closely linked, tightly linked. So what will be the result here? We get very less recombinants. Loosely linked, we get okay recombinants. Okay, that we'll try to understand here. So that two things. So where is our example? Closely linked, this one. Loosely linked, this one. This diagram explains loosely linked. This diagram explains closely linked. Okay, moving on. So what is the criteria here? What is the criteria? Let me take that particular lines from the textbook which explained about the crosses. That also we have to bring. People won't focus on this actually. That's the problem. Let me bring here. So first of all, he is saying about the cross A. Cross A. Cross A shows crossing between Y and W. Y and W. Cross B shows W and M. That here dominant wild type allele represented with wild sign. One more confusion. Generally, we show capital letter and small letter for dominant and recessive. Here they are shown in the form of plus is the dominant. That means here Y plus is the dominant, Y is the recessive. W plus is the dominant and W is the recessive. That is the another criteria you have to understand. And sir, why there is no gene here? This is X-linked, this is X, this is Y, and it is the X-linked one. Gene associated with X chromosome is called X-linked. So that also included here. That's why the confusion will come. Okay, ignore them. Ignore. Just we learn X-linked hemophilia, color blindness separately. No need to learn from here. So if any question comes, sir, there is no gene here. Okay, no gene. Why? Because they are the criteria. These two are the X chromosomes of the female. This is the X and Y of the male. So they want to show that. But you focus on this distance only now. Distance only. Okay. So Y plus is the dominant one. Small y, whatever the y. W plus is the dominant one. W is the recessive one. So if you understand this, I am erasing this. Now I focus on the cross. cross A, cross B. The strength of the linkage between Y and W is higher than WM. This line, the strength of the linkage between Y and W higher than WM and M. So see this distance. Distance is more loosely linked. Distance is less closely, tightly linked. You can say that was the line. Now let me show this cross to you. Let me show the cross to you. Capital Y, sorry, this one, capital, okay, because more adapted for that Mendel. YY, small YY, WW. This part I took, this part. This is yellow, white. I am writing yellow, white. Okay. And this cross, Y plus W plus. That is an X chromosome. That's why it is shown like that, X chromosome. So Y plus W plus. That's a single hemizygous condition we can say. This is in male. This is wild type. Wild. 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 Wild type for yellow, wild type for white. In that way. Okay. Then what will come in the first generation? Wild will come. Okay, wild will come. So that only shown here. Wild came, F1 generation, wild came, and small YW. What YW? Yellow and white. This is wild type, and next in yellow and white came. Wild and yellow white came. Next, actually, no need to understand, but people looking towards this, they always confuse this particular area because it is X-linked is also included here. X-linked. So if in our pattern, if you want to do, you have to do like this. Capital Y, Y by WW means hemophilia and color blindness combinations will do in general, isn't it? In that way, we have to do like this. Okay, shall we do like that? Shall we do like that? Okay, you are more comfortable with that, doing in that method. Let me draw Y, W, yellow, white. X-linked it is, yellow, white. Crossed with. So these kind of explanation, little they confuse people, don't know about hemophilia and color blindness. So I am revising for you. You are very champions in hemophilia and color blindness. This will be easily understandable for you. The earlier method is for the beginners who don't know hemophilia and color blindness. Now, as usual, X we have to take out. X, what we get? W, we should bring out whatever we got. Y plus X, Y plus, and W plus. Okay, so wild. Dominant character is wild. The another one I am taking X chromosome, then X with this X. So Y, W I am writing. I hope you don't be confused over if I do like this. Because we did so many combinations of hemophilia and color blindness. Now, Y. For Y, this X, and I am not drawing the table here. WY, I have to write one of the mother X chromosome. WY, I have to write one of the another X chromosome. So, what is WY? Yellow, white came. Male with yellow, white. You see here. This is XY. This one. I just shown in the form of visual like this. Yellow, white. And this condition in both Y, WW plus, WY plus. WW plus and YY. Okay, I have a problem, I think. WY. I should take, let me, wait. Should have used colors to understand. WY mother, one X chromosome. Here, W plus Y plus. And WY of one X chromosome, W plus and Y plus. This is a female. Something is happening. So then, okay, so to understand this phenomenon, if you want, you can draw, you can write like this also. But actually what they want to say distance between these two genes, YW, distance between the WM, there is a difference. This is loosely linked and this is tightly linked. What is the result? What is the result? In the previous page, we have seen that when I said loosely linked, parents will come more less recombinant. When they are tightly linked, more and more parents, very, very less recombinants. Okay. So that phenomena, if you watch, that will be enough. Just I am showing here, not a bigger diagram, it is X-linked hemophilia, color blindness angle. If you think it seems to be very simple, I want to prove that. You see recombinants are 1.3%, parental are 98.7. Why, sir? If you are able to answer, that means you understand the phenomena. Why, sir? Why? Because this curve, genes distance and the same chromosome, both are linked. Linkage is there. Genes are present on same chromosome, very closely linked. That's why we got very less recombinants. Okay, and you see here, parental type 62.8, parental type, recombinant type 37.2. Why, sir? Why? Because distance between the non-allelic genes present on non-allelic W plus allele, WM or non-allele, non-allelic gene present on same chromosome are loosely linked. Hence, we got 37.2. Non-allelic genes present on same chromosome, we got recombinants only 1.3%. That's why we say they are closely linked. What is the third criteria? Third criteria I explained in the first page. Parental gametes 25%, 25, 50%. Parental and recombinants both are equal. Parents equals recombinant, that is Mendel. Mendel selected two characters are present on two different homologous chromosomes, hence no linkage. Non-allelic genes present on same chromosome, linkage. Linkage. But in Mendel, then selected two characters present on different chromosomes. Okay. So I'll give the climax for this. That let us understand some phenomena like this. This is the first chromosome of human being, and the orange one I am taking, ninth chromosome of human being. This is the ninth chromosome. So with the known examples only I am going for. First chromosome is having RH factor. And the ninth chromosome is having A blood group. Character for blood group is there on ninth chromosome. Character for RH factor is there on first chromosome. So what is this? This is Mendel. Selected two characters are present on two characters. One is blood group and other is RH factor on different chromosomes. So what do you say, sir? No linkage. No linkage. Okay. We know that sickle cell anemic chromosome. In which chromosome it is present? Sickle cell anemic gene is there on 11th chromosome, isn't it? Sickle cell anemia is there on 11th. You know beta thalassemia is also on the same chromosome. So what do you say this is? Linkage. Means I am not taking so many examples. I am taking example for only which are there in our textbook. If I go wider, little wider, I can say like this. This is the first pair of chromosome. All 2,968 genes which are present on same chromosome are called linked genes. That pattern is called linkage. Sir, what about recombinants? If I take this A and Z loosely linked, but if I take A and B closely linked. So, if I take second chromosome, a character from first chromosome, a character from second chromosome, second pair, that is non-linked. This is Mendel. If I take one character from one chromosome, other character from other chromosome, that is Mendel. Independent assortation possible. If I take same genes on one chromosome very close, the third pattern we learned earlier, or same chromosome to distant genes, fourth pattern. So that means all these are issues are there in human being. All these are issues. Sir, how do I come to know? I will give the conclusion for that. How do I come to know? If parent equal recombinants, parental gametes, recombinant gametes, gametes angular, we are learning this. Parent and gametes are equal. I can say selected two characters are present on two different chromosomes, selected two characters genes. I am taking for our comfort always same criteria I am taking, capital Y, capital R, small r. This is Mendel. Here there is a possibility of independent assortation of the chromosome. This is the conclusion I am giving. Conclusion I am giving here, independent assortation. If parents more, recombinants less. Parents more, recombinants less. Here onwards linkage starts. Means I should draw only one set of chromosomes, and our selected two genes, two characters present on the same chromosome. Capital R, small r. Okay, so this is linkage. Okay, more parents will come here, less recombinants. More parents, less recombinants. For that, this percentage 98.7, 1.3 is there, 62.8, 37.2. So I am taking for this. The second one, that 62.8 and 37.2. More parents, how much sir? 62.8. Recombinant 37.2. Next, if parents are maximum, very less recombinants are present, then even phenomena called linkage, very less recombinant, that I can say like this. Our selected two characters present on same chromosome and very closely linked. That you have to understand. Same letter I am taking for easy understanding. For that, which one will be suitable? You see 98.7 parents, recombinants are only 1.3. So that I will write here. 98 parents are maximum we said. Enta 98.7% parent. Where if they are tightly linked, very less recombinant, 1.3%. So this is the linkage versus independent assortation. In this case, first one is independent assortation. What is the requirement for independent assortation? Selected two characters should present on different chromosome. What is linkage? Selected two characters present on same chromosome. If they distantly arrange, if they very closely arrange. That is the linkage 2 and 3. Here in the given, 2 and 3 are linkage. The one is independent assortation. So this is the complete story of linkage versus recombination. Linkage versus recombination. Okay, this is the story.